Hey guys, this video is to go over the note sheet that's titled Blood History, Basics, and Blood Typing. So if you haven't already collected those at the front of the room, make sure you get them. Okay, so let's talk a little bit about expectations. I expect that when I come back, or when I see you guys next week, that you will have all these notes done, and they will be properly written down, and that you will have completed your activity packet that goes with this topic. I expect that you will be fairly quiet, that you will be efficient, and you won't be giving any trouble or doing anything out of the ordinary, because I am not reteaching this to you. This is me teaching you through this video. You will have someone, one of your classmates should be up here running the video. I'm not going to do a lot of wait time in each, like from slide to slide. So at the end of every slide, I'll pretty much say, if you still are writing, then this person needs to go ahead and hit pause, so that you can pause the video throughout so you can get everything down. But I'm not going to be waiting for you to do it while I record this video. All right, so hopefully that was simple. Once the notes are done, you will use the notes to help you with the packet that I left, and this stuff goes over the genetics and the inheritance of blood. And I expect it to be turned in as you are leaving class today. So I tried to bold the words that, or the phrases that I want you to write down. This slide I missed them, so I'm just going to underline them for you really quick in red. And then we'll go ahead and start talking about this slide as soon as I change back to my laser pointer. Okay, so blood is a tissue, and it's made up of two sections. Some parts of it are solid, and other parts are liquid, or actually one part is liquid. The solid parts can either be red blood cells, also known as erythrocytes, white blood cells, also known as leukocytes, and platelets, which are just pieces of cells that have broken up, and they help your blood to clot and to form scabs so that you don't bleed out whenever you get cut. On your notes, go ahead and underline the word erythrocyte. Okay, that's the biological name for a red blood cell. And then also underline the word leukocyte for white blood cells. And then in the corner, like right here on your note page, I want you to write in the abbreviations for red blood cells, excuse me, and white blood cells that you might see me use or see used in other material. So red blood cells are abbreviated, whoops, sorry, let me switch to my pen. Red blood cells are abbreviated RBCSs, so RBCs, and make sure that the R, the B, and the C are capital. Whereas white blood cells are abbreviated WBC with a little s. Okay, so you might hear me refer to RBCs and WBCs. I am just talking about red blood cells and white blood cells. So in terms of its function, your red blood cells are primarily responsible for transporting gases like oxygen and carbon dioxide in and out throughout your body, so that you can recycle it and get what you need so that your system can function. Because of their duty of being carriers, they actually don't have a nucleus. If you look at this picture right here, you see how they're kind of sunken in the middle. It's because they're missing a nucleus. They have no genetic material. Your white blood cells are all about being part of your immune system and helping to protect you from foreign pathogens and foreign invaders that could potentially do you harm. And like I said before, your platelets are part of that clotting mechanism and that scab forming mechanism that your body utilizes so that when you get cut, you don't bleed out. Okay, now I am not going to build wait time into this presentation. So at the end of every slide, if whoever is running this for, my 3 classes, if there are still people writing, then just go ahead and pause the video until everyone's ready to move forward as a class. That being said, you guys need to be quick and concise with your writing. Make sure that you're staying on top of it, because the person who's running the video shouldn't have to wait for 5 minutes on every single slide for you to get done. Okay, that being said, you should be done. Moving on. So if you need to pause, this is the time at which you should pause the video. All right, so this part goes with that little table that you have under the section that says blood history, and the only dates that I pulled are the ones that are most important as it applies to forensics. So starting in the early 1900s, a gentleman by the name of Castle developed the first presumptive test that we use to determine the presence of blood. So you need to know the date, the person, and what they did. Then in 1901, Carl Landsteiner discovered that there are actually 3 blood types. At least at that point in time, he thought there were 3 blood types. So he found the blood types A, B, and O. In 1902, this guy discovered that there was an additional blood type in the form of blood type AB. Then in 1904, Carl once more made another discovery, which would be the rhesus protein or rhesus factor, which helps to give your blood either that positive or negative connotation. And in 1979, Dr. Bloomberg discovered a method to detect antibodies, or he developed a method to detect antibodies, which we still utilize to this day when we're trying to tell the difference between human blood and animal blood. So I know this particular slide you're going to need some time because you are filling all of this information in. Please note you are responsible for every date, every person, and all of their contributions on this particular slide. There are only 5 of them. So now would be a good time for you to hit pause and get that table filled in so that you can move on. So pause if you need to. Alright, so let's get into some of the nitty-gritty. Now, your body has the ability to tell the difference between self and non-self. That just means that your body, like your actual cells, can figure out other cells that have been made by your body versus cells that came from the environment. So like bacteria or viruses or any kind of pathogen or cell that doesn't belong to you, even like a cell that came from a different person's body. The way they do that is because most cells, and especially red blood cells, carry proteins on the surface, on their cell membrane. They have these proteins that are embedded. Now, the presence or absence, and it's important that you realize that it could be either of these, so either the presence or the absence of these proteins is kind of what determines which of the 4 blood types you will have. These proteins are called cell surface proteins, but they can also be called antigens. So antigens, cell surface proteins, they're all the same thing. All right, if you need to pause the video, now would be the time. So there are 2 major antigens. They are called antigen A and antigen B. And these are the ones that pretty much determine the blood type that you are going to have. If a person has antigen A on their blood cells, then that person has type A blood. If the person has antigen B on their blood cells, that person has type B blood. If the person has both antigen A and antigen B, then that person has type AB blood. And if the person has neither antigen A nor antigen B, the person has type O blood. So it's pretty self-explanatory. If you have protein A, you have type A blood. Protein B, you have type B blood. If you have both protein A and protein B, you have AB blood. And if you don't have either of them, you have type O blood. All right, so you need to fill out everything in bold and give you a little bit to do that. If you need to pause the video, now would be the time to do that. All right, so that takes care of the A, B, O, or AB. Now we need to talk about the positive or the negative. And that is controlled by what we call the Rh protein or Rh factor. About 85% of people make an Rh protein, which makes their blood type positive. So it makes them Rh positive for whatever their blood type is. The other 15% of the population does not make this protein, so that makes their blood type negative, or Rh negative. So when we talk about someone having A positive blood, that person makes antigen A, so they have type A blood, and they make the Rh protein, so that's why it's positive. But if we talk about someone having type A negative blood, then that person still makes the antigen A, which is why their blood is type A, but they do not make the Rh, which is why they have that negative sign. So the positive or negative is controlled by the Rh. The type of blood, A, B, O, or AB, is controlled by those proteins A and B. in either having them or not having them. All right, so really quick, if we had to break it down, this is where the U.S. population would fit in terms of how many people have what blood type. So the most popular blood type, or the one that's the most abundant, I should say, because it's not popular because we can choose what we want, but the one that's most abundant would be type O blood with 43%. Type A blood runs a close second at 42%. Note that type AB is the least common, and that's at 3%, and type B is at 12%, so it's kind of in the middle of the pack. So earlier I mentioned that your white blood cells are part of your immune system. They're there to protect your body from foreign material that could potentially hurt you or make you sick. One of the ways that they do this is by making a molecule called an antibody. So what antibodies do is they have the ability to connect to other kinds of cells and cause those cells to connect to each other. So I think of antibodies as being handcuffs. They kind of handcuff all the bad guys and keep them in the same location so that the rest of your white blood cells can come in and get rid of them. Now, in the presence of certain proteins, antibodies are going to cause cells to clump. Now, just like how we have two kinds of antigens, we also have two kinds of antibodies. We have antibody A and antibody B. I want you to note that antibody A clumps with antigen A, and antibody B clumps with antigen B. Okay, so again, there are two kinds of antibodies, antibody A and B, and they clump or get together, connect together with certain kinds of antigens. Antibody A clumps with antigen A, antibody B clumps with antigen B. But this kind of creates a problem because earlier we learned that we have blood types based on antigen A and antigen B. People with type A blood have antigen A. People with type B blood have antigen B. People with both of them have AB blood, and people with neither have O blood. So if I make antigen A and I make antibody A, I could have a situation that's not good. I could have blood clumping as it flows through my veins. That's not a good thing. So what your body does is it makes sure that you make the opposite antibody to the antigen that you have. So in other words, if you have type A blood, you make antibody B. If you have type B blood, you make antibody A. If you have AB blood, you don't make any of those antibodies, neither A nor B, because they would clump regardless of what you made. If you have type O blood, you can make both antibody A and B because you don't make any of the antigens. So this is your body's answer to a potential problem. All right? It is very important that you remember this section that we've just gone about, how your body avoids clumping. It's by making the opposite kind of antibody to the ones that you present on your red blood cells. Okay, if you need to pause the video, now would be the time. All right, so this is the same image that you have. It just goes over, it's a visual for the words that we pretty much just talked about. So if we look at it, these are our 4 types of blood. This person has type A blood because they make A proteins. This person has type B blood because they make B proteins. This person has type AB blood because they make both A and B proteins. And this person has type O blood because they make no proteins, neither A, nor B proteins on their blood cells. If you have type A blood, because you have the A protein, it means that your body makes the B antibody. If you have type B blood, because you have B proteins, it means that your body makes the A antibody. If you have type AB blood, you have both A and B proteins. Your body doesn't make either antibody A nor antibody B, because you have both of those proteins on your red blood cell. If you are type O, you don't have antigen, A or B on your blood cell, so your body makes both antibody A and antibody B. All right, so what's the actual blood typing? Like, how do we figure out our blood type? Well, we do it by running 3 very simple tests. And all that blood typing is, it's a way to identify and to match different blood samples together. So you will take a little depression jar, or well, sorry, not jar, but depression wells, and you will put a sample of blood in each depression, and then you mix it with different reagents. So you're going to do a protein test for protein A, another test for protein B, and a test for Rh protein. And depending on which one's clump, it tells you what kind of blood type and if that person is positive or negative. Just so you know, this first picture that I'm circling here, this would be an example of non-clumping, so just regular looking blood, whereas down here, this picture that I'm now circling is an example of clumping blood. If you look at it, you can see how the red parts have kind of moved together, closer together, and then you can see some like white spaces in between. So this would be clumping, and this would be no clumping. So let's talk about the genetics part of blood typing. Now, the gene that controls blood type is autosomal, which just means that it's found on a body chromosome, not on a sex chromosome. So it's not on your X chromosome or your Y chromosome. It's actually found on chromosome 9. And it works in a very specific way. So whenever we talk about genetics and genes, you should expect to hear the words dominant and recessive and homozygous and heterozygous being thrown around. And I will review you on what some of those terms are a little bit later on. So you need to remember that A and B blood groups are dominant or stronger or more likely to be seen than O blood groups. So these guys are stronger than this guy. But A and B, when you compare them to each other, they are what we call codominant. So it means that they are equally strong. A is just as strong as B. So if they're both found in the same person, there's a chance that that person will have AB blood, for example. A won't overpower B and B won't overpower A. You have to also remember that the way genetics works is that you get 2 copies of your genes from each parent. You get one from your mother and one from your father. So you can get an A and another A from your mom and dad and have type A blood, or you could get an A and an O from your mom and dad and still have type A blood. And we'll talk about how that works in a little bit. All right, so write down what you need to. If you need to pause the video, go ahead and do so now. Right, so this is another slide where I forgot to bold the words, so I'm just going to underline. You are writing down the terms. In here, I just remind you of what those words dominant, recessive, and codominant mean. So in genetics, dominant just means that this is the gene that you are going to express. This is the trait that you are going to express, and it's normally represented by a capital letter. Whereas recessive means that this is a trait that is not as easily expressed. It is represented by a lowercase letter normally, but for you to have a recessive trait, for you to be able to see a recessive trait, it means that both genes that you get from your mom and dad need to be recessive. So let me give you an example. People who have blue eyes would be an example of a recessive trait that is controlled by 2 recessive genes. Blue eyes are not dominant. They're not the norm, okay? If you have blue eyes, it's because you got that blue eye gene both from your mother and from your father. And if there's anyone in the room with blue eyes, I want you to think about it. Chances are both of your parents don't also have blue eyes. You might have one parent with blue eyes, and you might have another parent with green eyes or brown eyes or some other color eyes, or you could have both parents that don't have blue eyes at all. They have a different color to you. But the only reason that your eyes are blue is because your parents are heterozygous for whatever their eye color is. So it means that they have a gene for brown, let's say, and a gene for blue. and they happen to pass on their blue-eyed gene to you. Both of them did. That's the only reason your eyes were blue. So let's say if your dad had a blue-eyed gene and a brown-eyed gene, and he passed on his brown-eyed gene, and your mom passed on her blue-eyed gene, your eyes would be brown because you need both genes to be blue-eyed. That's how recessive genes work. So in the case of O blood is a recessive trait. The only way you can have blood type O is if you got an O gene from your mother and your father. If you get an O gene and a different kind of gene, an A or a B, then you are not going to have O blood. You have to have both recessive genes for that feature to show. And again, like we said, codominant just means that one gene is not stronger or does not overpower the other. Both of them are seen. So in the case of people with AB blood, A isn't only seen because A isn't stronger than B. B isn't stronger than A. They both share how strong they are. They're equally strong, so you have both of them. Okay, so this is a representation of a giant Punnett square. And I don't know if you remember how to do Punnett squares because it's been a while since you guys have had biology. So I'm going to go ahead and try and refresh your memory real quick. So I'm going to change pens. Now the way Punnett squares work, or the way I used to teach it to my freshmen would be to start over here on this left-hand side. Anything on the left needs to be written in every box to the right. So this A needs to be written all the way on this side. So like A, and A. This B, same thing, written all the way across. B, B, B. And this O, same thing, written all the way across. O, O, and O. Now moving to the top and working our way down, everything on the top needs to be written in every box below it. So A written in every box below would give us A, and A. B written in every box below would give us B, and B. And O written in every box below would give us O, and O. So you should be seeing a mix of letters right now. Like right here we have big A, and then we have big A and O, and big A and O. Well, big A, and big A and O are both going to be A blood, like A blood type. Sorry if that ran out of room. Then over here where you see like B and O, and B and B, both of those are still going to be B blood type. So realize that there are two ways that you can have A blood type or two ways that you can have O blood type. But I want you to pay attention to the fact that there's only one way to have AB and only one way to have O. Now really quick, BA and AB, exact same thing. It's like saying 3 times 5 is 15 and 5 times 3 is 15. The outcome is going to be the same even if you flip the numbers around. So in genetics, even if you flip the letters around, BA is still technically AB blood type. All right, so I'm going to switch to, you might have been filling this out with me as I was going, I don't know, but I'm going to switch to the version where it's filled out in like text material as opposed to my awful handwriting. So moving on to the next slide, which is pretty much this slide just in neater handwriting. All right, so this is exactly what we got when we did it. It's just all nice and neat now. So go ahead and fill out that that Punnett square if you haven't already done that. Okay, so on this one you're going to have to write and pay attention along with me as I go. Now you should have this version of the Punnett square on your assignment sheet, but when you work problems for me, the problems are sometimes like word problems. So I might tell you that a parent that is homozygous dominant with a parent that is heterozygous ask. Okay, so one of the things you're going to have to learn how to do is how to process a crime scene that involves blood, because this will be one of the units where we do a crime scene. There are 3 steps to doing this. The first being that you have to confirm that the stain that you are looking at, that red spot that you are looking at, is actually blood. There are 2 tests that will tell you if you are working with blood or if you're working with something else. The first one is called the Kastle-Meyer test, where you take a solution, you spray a little bit of it on your sample or on a sample of blood, and if it turns a bright pink color, it tells you that blood is present. If it does not change color, then it tells you that what you're looking at isn't blood. It's something else that has a red color to it. The other test is called the leukomalachite green test, and it's the same concept. You spray a little bit of the solution on your sample, and if it's actually blood, it will turn a bright green color. Once you know that the stain you're working with is actually blood, the next step is to confirm that it is human blood and not some kind of animal blood. So this is done through the ELISA test. So that is the antibody detection development that we talked about under the history of blood. So the way it works is you take a sample of human blood and you inject it into a host animal, and you leave that animal alone for a little bit. The animal will naturally make antibodies against that blood because that blood did not come from that rabbit or that mouse or whatever it is you're using. You can then extract those antibodies by taking some blood from the animal, taking everything else out and leaving just the antibodies, and then mixing the antibodies in with what you think is the human blood you found at your crime scene. Because the antibodies are human antibodies, they should cause the blood to clump together if the blood is from a human being. If that blood comes from an animal, however, it will not clump after you mix it with the antibodies. It will just look like regular blood, like nothing special or different would have happened. So clumping tells you that it's human blood. No clumping tells you that it would be animal blood. All right, and then the last thing, that you would do once you know that you're working with human blood is to figure out the blood type, and that you do that by doing your 3 blood typing tests. The test for protein A, the test for protein B, and the test for the Rh factor. Okay, so this is pretty much the last of it. I just want to quickly review some genetics terms with you. So I'm just going to write them out in my less than stellar handwriting. And hope that you are going to be able to copy these down. You should have some space underneath this last section on your note sheet. So let's talk about what homozygous means or looks like as it refers to blood groups. So if something is homozygous, the word homo means same. Okay, not anything else that you might be thinking. We're just going to take it as it means the same thing. So same, whereas heterozygous means different. Okay? And it is talking about the actual genes. So we're talking about the letters. So in this case, same letter. Okay? So that could be 2 capital letters or 2 lowercase letters. Both of those would be the same. Hetero, again, when we refer to the letter, just means that one is going to be uppercase and normally one would be lowercase. So the letters are different from each other. All right, so I'm going to give you a second to copy that down. If you need to pause the video, feel free to go ahead and do so, and I'll keep going on with a new slide. Okay, guys, I just remembered that I can actually type on the iPad, so you will not have to be subjected to my less than stellar handwriting. So these are some terms that you would have learned your freshman year: homozygous dominant, heterozygous, and homozygous recessive. let's talk about how that applies to blood groups. So homozygous dominant, remember homo means same, dominant means that they're going to be capital letters, the same kind of capital letter. So homozygous dominant genes for blood type would be, for example, big A, and big B, big B. This would give you type A blood, and this would give you type B blood. Okay? An example of heterozygous blood would be, again, the letters are going to have to be different because heterozygous means different, but in this case it would be A and O or B and O. A and O still gives you type A blood. It's just heterozygous. You got that O from one parent, but remember, A is stronger than O, so A is going to be the blood type that you have. B and O would still give you type B blood because of the same situation. B is stronger than O. It will overpower O. So even though you got that O gene from a parent, because you got that B gene as well, you're still going to have type B blood. Homozygous recessive would be, normally it's 2 lowercase letters, like in any other kind of genetics, but when we're dealing with blood types, our O is actually recessive. So the only way you can be homozygous recessive is to have 2 O's, O and O, and that person would have O blood. Okay, the only one that we've left out would be AB, but remember that AB is codominant because they both share strength equally. All right, so hopefully this wasn't too painful. The expectation now is that you will take the notes that you've been given and you are going to work on the genetics packet. I expect all questions to be answered. I expect all Punnett squares to be filled in. If you feel like you need to add another Punnett square, if you need to attach another sheet of paper to show you're working so that you figured out these questions, all of that is fine. But I expect it all to be done with your names on it and turned into the box before you leave class. So you have the remainder of time to finish that up. All right, I will see you guys next week. For Friday's lesson, it's going to be the same thing, pretty much the same concept. You're going to have a video, and then you're going to have some practice. All right, guys? So be good. I might pop in and check on you. Take care. Bye.